Welcome to this lecture on reproductive biology. Today, we are exploring one of the most fascinating and critical events in early mammalian development, the maternal recognition of pregnancy. Imagine a biological clock ticking down to zero, where a tiny embryo must send a life-saving message to its mother to survive. This invisible communication happens in the first few weeks of pregnancy and is the ultimate biological save mechanism. Let us dive in and understand how this incredible process works. To understand why this process is so important, imagine you have a monthly subscription box that automatically cancels itself unless you explicitly send a text message saying, "Keep it coming." The female reproductive system works in a surprisingly similar way. Every month, the uterus prepares a rich, thick lining to host a potential baby. However, this lining has a strict expiration date. If a fertilized egg or embryo arrives, it must urgently send a chemical text message to the mother's body before that expiration date hits. If the message is received in time, the lining is kept intact. If not, it is shed and the embryo is lost. Now that we understand the urgency of this message, let us look at the scientific mechanism behind it. Maternal recognition of pregnancy is the physiological process where the developing embryo signals its presence to the maternal endocrine system. The primary goal here is to prevent luteolysis, which is the programmed destruction of the corpus luteum in the ovary. The corpus luteum is a temporary gland that secretes progesterone, the master hormone responsible for keeping the uterine lining stable. By sending specific biochemical signals, such as human chorionic gonadotropin in humans, the embryo rescues the corpus luteum from its natural death, ensuring a continuous supply of progesterone. Having discussed the biological players, let us visualize this life-saving communication. On your screen is a schematic diagram of the female reproductive tract during early pregnancy. On the left, you can see the blastocyst, which is the early embryo, implanted in the endometrium of the uterus. Notice the dashed arrows representing the chemical signal, human chorionic gonadotropin, traveling through the bloodstream. This signal reaches the ovary on the right, specifically targeting the corpus luteum. The red cross over the word luteolysis shows that this destruction is blocked. As a result, the corpus luteum continues to produce progesterone, which travels back to maintain the endometrium and prevent menstruation. This visual pathway we just explored highlights why this concept is absolutely vital. It represents the most critical bottleneck in early pregnancy survival. In real life, a significant percentage of early miscarriages occur simply because this communication fails. The embryo might be perfectly healthy, but if its chemical signal is too weak or sent too late, the mother's body will initiate menstruation anyway. In reproductive medicine, such as in vitro fertilization, doctors closely monitor these exact hormones to confirm successful implantation. Furthermore, this very signal is what over-the-counter home pregnancy tests detect, making this invisible biological process something millions interact with daily. Knowing that the embryo is racing against a biological clock, let us look at how we can mathematically determine this absolute deadline. The formula on screen calculates the day of luteolysis, which is the exact day the corpus luteum begins to die. Conceptually, this is simply the day the egg was released, plus the natural lifespan of the corpus luteum, if no pregnancy occurs. DL represents the specific day of the menstrual cycle when luteolysis begins. DO represents the day of ovulation, when the egg was released. Finally, LC stands for the natural lifespan of the unrescued corpus luteum, measured in days. If the lifespan increases, the deadline is pushed back, giving the embryo more time. Before we move on, let us see this formula in action with a concrete biological timeline. We're given a standard 28-day menstrual cycle where ovulation occurs exactly on day 14. The corpus luteum is programmed to die 10 days after ovulation. We need to find the exact day luteolysis begins. We chose our deadline formula because it perfectly models this biological addition. We substitute 14 for the day of ovulation and 10 for the lifespan of the corpus luteum. Adding these together, we find that the corpus luteum will begin to self-destruct on day 24. This day 24 is the absolute, non-negotiable deadline for the embryo's chemical signal to arrive. Now that we have established the deadline, we must determine when the embryo can actually start sending its life-saving signal. This brings us to our second formula. Conceptually, the embryo cannot signal the mother until it has traveled to the uterus and implanted. DS represents the day the embryo successfully implants and begins secreting the hormone. DF is the day the egg was fertilized. TT represents the travel time it takes for the embryo to move down the fallopian tube and burrow into the uterus. If the travel time increases due to biological delays, the day the signal starts will be pushed later into the cycle, dangerously close to the deadline. Let us apply this second formula to our ongoing scenario to see when the communication actually begins. We know fertilization aligns with ovulation on day 14. We are also given that the embryo's travel and implantation process takes exactly 6 days. We need to find the day the signal starts. By substituting 14 for the day of fertilization and 6 for the travel time, we simply add them together. This shows that the embryo implants and begins sending the signal on day 20 of the menstrual cycle. Physically, this means that before day 20, the embryo is floating freely and cannot communicate. Once day 20 arrives, the biological text message is finally sent into the maternal bloodstream. We now know both the deadline and the start time, which leads us to calculate the breathing room the embryo has. This is known as the critical window. Conceptually, this formula finds the difference between when the signal starts and when the system shuts down. W represents this critical window of time, measured in days. DL is our ultimate deadline day for luteolysis, and DS is the day the signal starts. If the signal starts later, DS increases, which shrinks the window W. A smaller window means a higher risk of pregnancy failure, as the embryo has less time to produce enough hormone to rescue the corpus luteum. Let us bring our previous calculations together to find the exact margin of error built into the human reproductive system. From our first example, we found the deadline is day 24. From our second example, the signal starts on day 20. We are asked to find the window of time available. We subtract the start day, 20, from the deadline day, 24. The final calculation gives us a result of 4 days. Physically, this means the embryo has a critical 4-day window to produce enough hormone to reach the ovaries and halt the destruction of the corpus luteum. If the journey were delayed by just a few days, this narrow window would close and the pregnancy would be lost. Once that 4-day window opens and the signal begins, doctors need to verify if it is strong enough. They do this by tracking the mathematical model of hormone production. Conceptually, once the embryo implants, the concentration of the signal hormone multiplies rapidly in the mother's blood, following an exponential growth curve. In this equation, C of T represents the hormone concentration at a given time, measured in milli-international units per milliliter. C0 is the initial concentration from the first blood test. The variable T is the total elapsed time between measurements in hours, and capital T is the doubling time. If the elapsed time increases, the overall concentration grows exponentially, provided the doubling time remains healthy. Let us look at a clinical example where a doctor is using this exact formula to verify if maternal recognition is happening correctly. A patient has her blood drawn on Monday at 8 in the morning, showing an initial hormone level of 50. The doctor expects a healthy doubling time of 48 hours. We need to calculate the expected concentration for her next test on Wednesday at 8 in the morning. First, we must determine the total time elapsed between the two blood tests. Counting the hours from Monday morning to Wednesday morning gives us exactly 48 hours. This value will serve as our elapsed time variable for the exponential growth equation in the next step. Now that we have our elapsed time, we can complete the clinical calculation. We substitute our known values into the exponential equation. Our starting concentration is 50. The exponent becomes the ratio of the elapsed time, 48 hours, to the doubling time, which is also 48 hours. Because these times are identical, the ratio simplifies beautifully to exactly 1. This means the hormone has gone through exactly 1 complete doubling cycle. Multiplying the initial 50 by 2 gives us a final expected concentration of 100. Physically, if the patient's blood test shows a level around 100, the doctor can confidently conclude that the embryo is successfully signaling the mother's body and the pregnancy is safe. We have covered the full timeline and mathematics of this incredible biological event. Let us reinforce the 3 most important ideas. First, maternal recognition of pregnancy is the ultimate biological save mechanism, where the embryo secretes a chemical messenger to prevent the destruction of the corpus luteum. Second, this process operates on a strict biological clock, leaving the embryo with a very narrow critical window to establish communication before the uterine lining is shed. Finally, we can mathematically verify this life-saving signal by tracking the exponential growth of the pregnancy hormone. Understanding this delicate communication helps us appreciate the fragility of early pregnancy and the science behind modern fertility monitoring.